Okay, good morning, students. Okay, yesterday the topic is microbes in human welfare. We are discussing the sewage treatment. Last we are discussing sewage treatment. The sewage treatment means it is the filtration of waste waters and drainage water. Okay, the filtration process means primary treatment, secondary treatment, and anaerobic tank. Then finally, the filtration of water will be through releasing for canals and rivers. Here, our Indian government has to be taken the Ministry of Forest and the Ministry of Forest and Environment. The Ministry of Forestry and Environment can take a action for Yamuna Action Plan and Ganga Action Plan to using bioremediation. Bioremediation means to using microorganisms to remove oils and other materials in rivers. Okay, that is sewage treatment. Okay, now we are discussing production of biogas. Production of biogas. Okay, we will see the diagram. Production of biogas. Okay, here it is to be filled. Okay, this is the digester. This is gas holder. Okay, this is, here it is to be, it is called using dung and water. This is called sludge. Okay, so production of biogas. So first we are using, it is dung and water to mixes. First dung and water mixes. Dung means animal waste. It is called dung, to mix water to pump in this tank. This is called a digester. Okay, here in this, what are the raw material will be mixed in this, directly this material will be pumped in this tank. Here that organic material will be digested to release high amount of methane and carbon dioxide and water of hydrogen. Okay, and hydrogen. Okay, so here how it will be digest means, here it is to be found, the most of the bacterium is called methanogens. That methanogens, it is a common bacteria, is called methanobacterium. Methanobacterium. Here it is to be found in cattle. Here it is to be found in cattle. Means, so here it is to be found the methanogen bacterium. Methanogen bacterium. Methanogen bacterium. Okay, methanogen bacterium. This bacterium is having, okay, this bacteria is having, okay, this bacteria has to be releasing methane. Here present in the cattle. That cattle is having this bacteria. Okay, here present in the cattle. This cattle is having rumen. This cattle is having rumen. This bacteria is having, this cattle is having this bacteria. This bacteria is present in rumen. Is a part of stomach. Means rumen is to be found in part of stomach. It is a part of cattles. Okay, here the rumen is having in bacterium, methanogen bacterium. This bacteria, methanogens bacteria. Methanogens bacteria. In this methanogens bacteria will be, occurred in cellulosic activity. In substrate cellulosic digestion activity. Okay, here in this methanogens will be the cellulosic material. Cellulosic material will be digested. Okay, cellulosic material will be digested. It is very important role play in the cattle. Because of it is to be supplying for nutrients. Supplying for nutrients. And then finally then after cellulosic material will be digested to play the important role of in nutrients to supply the cattle, and finally the waste is called the dung. This dung also called gobar. This is called a production of gobar gas. Means here the methanogen bacterium is to be found in this tank. Here the sludge will be digested. In the cattle. In cattle is having this type of bacteria. This bacteria is also to be present in rumen. Of cattle. Okay, rumen is a part of stomach in cattle. Okay, in rumen is having methanogen bacterium is there. That methanogen bacterium is where cellulosic material will be digested. Then finally it will be releases dung. The dung is called a gobar. After cellulosic material will be digested, it is play important role to supply the nutrients of cattle. Okay, then finally after digested, the gobar will be released. The waste material will be pumped into the another tank. Okay, this waste is also to be using for fertilizers. Okay, different types of farmers will be using in fertilizers. This waste fertilizers means it's like a organic farming. Okay, so using different types of crops to using the waste sludge. This is the biogas production. Okay, once again I will explain here. Okay, this is dung. This is dung and water will be mixtures. Dung and water will be mixtures. Dung means it is an animal waste. Dung means animal waste. So this material will be going to this tank. Okay, this tank. This tank here it is having, it is a digestive tank. Digestive tank means here it is to be found in cattle. Okay, this cattle and bacteria. Okay, these methanogen bacteria will be found in this tank. These bacteria is to be present in the cattle. Present in cattle means that cattle present in the cattle. That cattle is having rumen. That rumen is present in the bacteria in rumen. So here that rumen, the bacteria will be, cellulosic material will be digested. After, then finally it will be releases dung. That is called a gobar. Okay. Then finally the waste products will be going to another tank. This is called sludge. That is using for biofertilizers. It is using for organic farming. So this is the production of biogas. Okay. This bacteria is to be found in rumen of cattle. Okay, the methanogen bacteria is to be found in rumen of cattle. Rumen is a part of stomach in cattle. Okay, so stomach in cattle. So the cellulosic material will be digested. Then it will be play the important role of to supply the nutrients of cattle. Then finally the waste material is called dung. That is called a gobar. That is called methane gas. This is called methane gas in CH4. Methane gas. That high amount it will be released for gas. That gas will be directly connected in our houses. That gases will be directly connected in our houses or cooking rooms. Then using in biogas. Next, coming to the topic is microbes in biocontrol agents. Microbes in biocontrol agents. So one more thing we will discussing the biogas production. Okay, the biogas production will be conduct in our India. So IARI and KVIC means Indian Agriculture Research Institute, and one more thing is Khadi Village Industries Commission. The two departments are to be conducted in biogas production. Okay. Next, microbes in biocontrol agents. The first one is ladybirds and dragonflies. First one is ladybirds and dragonflies. Okay, dragonflies to removing aphids and mosquitoes. Means microbes in biocontrol agents means, so we are using different types of microorganisms. It is also called organic farming. Okay, so different types of microorganisms we will use to remove harmful organisms or harmful insects. Okay, to prevent diseases also. Okay, aphids and mosquitoes will be in there in some different types of crops. Okay, we are using ladybirds and dragonflies microorganisms to remove aphids and mosquitoes. Okay, to use ladybirds and the dragonflies microorganisms to remove aphids and mosquitoes, we are using in where it is using in agriculture and organic forming. Next thing is Bacillus thuringiensis. Bacillus thuringiensis. Bacillus thuringiensis. It is called a BT. It is called a BT. It is called a BT. BT means, so this Bacillus thuringiensis is modified, is genetically it is modified. It is coming from through biotechnology. It is coming from through biotechnology. BT means it is genetically modified microorganism. Genetically modified microorganisms to remove butterfly caterpillars. Butterfly caterpillars. Butterfly caterpillars. To remove butterfly caterpillars. Okay. So Bacillus thuringiensis is a BT. It is called genetically modified organism or genetically modified gene. So it is to use to remove butterfly caterpillars. Butterfly caterpillars means Bacillus thuringiensis. It is coming from through biotechnology. It is genetically modified thing. This is called Bacillus thuringiensis, to use remove for butterfly caterpillars. Butterfly caterpillars. Next thing is called BT cotton and BT brinjal. 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 Okay, these are fully genetically modified. Fully genetically modified means through using biotechnology. Through using biotechnology to insert gene. To inserted gene. Inserted gene means the ingestion of gene or ingestion of microorganisms. Ingestion of gene or microorganisms to be protecting for insects and pathogens. For in case the harmful microorganisms or harmful insects will be fall in crabs. Okay, that's the time they will be eat some tissue. So that tissue is having BT cotton and BT brinjal means it is a inserted gene. It is a inserted gene. So the chemicals will be influenced particular insects or whatever it is. The chemicals will be influenced to kill that insects. Means it is genetically modified plants. It is genetically modified plants means BT cotton and BT brinjal. So it is inserted new gene for the plants means BT cotton and BT brinjal. So they have pathogen resistance power. Pathogen resistance power is having in these plants means genetically modified plants. In case the harmful organisms will be enter to eat the tissue. Okay, there is having BT cotton and BT brinjal. Okay, these chemicals will be influenced to kill that microorganism. Insects, harmful insects and whatever it is. Okay, birds also. Okay, next is called trichoderma. Next thing is called trichoderma. It is called fungal membrane. It is to be found in root ecosystem. Means trichoderma, it is a fungal membrane. It is to be protecting for root ecosystem. Okay, so whatever the pathogens will be enter and kill the root system. So in this trichoderma, microorganisms to be protect and to kill the pathogens. In most of the time, the trichoderma will be found in root ecosystem. Trichoderma will be found in root ecosystems. Okay. The next thing is baculovirus. Baculoviruses. nucleoviruses. Okay, it is also called nucleopolyhedroviruses. It is also nucleopolyhedrovirus. Okay, this is a species found in glomus. Baculovirus is species found in glomus. It is also called nucleopolyhedroviruses. Baculovirus is also called one of the pathogens. Okay, the baculovirus is also one of the pathogens. So in these pathogens, okay, who are, okay, so which organisms are useful the plants to conserve that insects and birds. So which are harmful insects and birds will be there to kill the baculoviruses, Baculoviruses also to be pathogens. Okay, so which are useful insects and arthropods and different types of microorganisms will be there. That what are the useful to the plants, so they will be conserved in these baculoviruses. Okay, so what are the harmful in the plants, they will be killed. So baculovirus is one of the pathogens. It will be insects and arthropods. Okay, it will be kill insects and arthropods. Okay, insects and arthropods will be killed. They are harmful in the plants. And what are the useful to consider and to conserve it? Okay, they are another thing is harmful. There any harmful will be present in the plants, they will be killed. So this aid is called IPM. IPM means integrated pest management. Integrated pest management means whenever it will be available, means harmful will be there in the plants, that time it will be baculovirus to be act pathogens to kill that one. In case they are useful to plants, okay, there is no reaction. They are useful because of they are useful for plants. So baculovirus also, it is called a pathogens. Okay, it will be kill insects and arthropods. They are useful organisms will be there in our plants. They are conserved. Okay, this is microbes in biocontrol agents. Microbes in biocontrol agents. Once upon a time, the farmers also to be using this all types of microorganisms. Not all types. This is the genetically modified things. Means through coming from biotechnology. So using organic farming in this microorganisms. Next, coming to the next thing is biofertilizers. Next thing is called biofertilizers. Which are biofertilizers? Microorganisms are using in biofertilizers. The first one is bacteria. The bacterium is to be found. It is called rhizobium. Which type of bacteria? Rhizobium bacteria. It is to be found in rhizobium bacteria. So rhizobium bacteria and agidospilum and azotobacter. Agidospilum and azotobacter. The bacteria are to be found in rhizobium and agidospilum and azotobacter. Okay, the rhizobium is the family of leguminous The family of leguminous. Okay, it is also to be found in symbiotic bacteria. Symbiotic bacteria. The symbiotic bacteria means it is the relation between two organisms. Means the mutation between two organisms. Mutation between two organisms. It is also to be found in symbiotic bacteria. So this is also Azospilum and Azotobacter. So they are found in root. They are found in root system. They are found in root system. To be roots are to be found in nitrogen fixation. Nitrogen fixation. Okay, it is to be helping of the nitrogen fixation on the roots of plants roots. Okay, they are present in the roots. Okay, these bacteria, Rhizophora, Aspergillum and Azotobacter is to be found in the roots. Means it is the fixation of nitrogen. It is fixation of the nitrogen. The collection of nitrogen, it will be supplying for the throughout the root system. And what are the pathogens are to be entered in that root system, they will be killed, and what are the nutrients and supplements of water and whatever it is, they are receiving and supplying for the roots. Receiving and supplying for the roots. They are bacteria, Rhizobium and Aspergillum and Azotobacter, they are present in the root system. It is to be nitrogen fixation. They will be collected and supplying for nitrogen. Nitrogen nodules, like nodules like. Nitrogen is a nodules. Okay, small nodules is there on roots. Because of the bacteria will be collected nitrogen nodules to be supplying for the plant, supplying for the plants. Whatever it is, organic material from comes to soil to plant, it is to be receiving and supplying for the plant. Next thing is called fungi. It is a fungal membrane. It is a fungal membrane. Here it is to be found in mycorrhiza. Mycorrhiza fungus. Fungi means mycorrhiza. The mycorrhiza is divided into endomycorrhiza and ectomycorrhiza. Ectomycorrhiza and endomycorrhiza. Okay, the ectomycorrhiza is to be protecting for the root epidermal layer. Roots epidermal tissue. Okay, the ectomycorrhiza is to be present in the epidermal tissue in the roots. So it is to be prevent the pathogens. It is to be prevent the pathogens. What are the organic raw material? It is to be exchanging in the epidermal tissue. Epidermal tissue. And the endomycorrhiza is to be found in xylem. Okay, found in xylem. So what are the these cortical cells? Cortical cells. Okay, it is to be exchanging of the nutrients and supplement, water and minerals. Water and minerals. Water and minerals and nutrients. Okay, it is to supplying for in the xylem. It is to be found in endomycorrhiza is to be found in xylem. Okay, ectomycorrhiza is to be found in root epidermal tissue. Root epidermal tissue. Epidermal tissue, it is to be protecting from pathogens, and what are the organic materials and organic supplements to be soil to plant, it is to be supplying for the epidermal tissue in ectomycorrhiza, and endomycorrhiza, it is to be found in inside this called xylem in cortical cells. So what are the important things in water and minerals and nutrients will be supplying for the plants, Next, the last thing is called cyanobacteria. Cyanobacteria is having nostoc and asclerotorium, next anabenum. Okay, this cyanobacteria. This is called cyanobacteria. This bacteria is to be found in nitrogen fixes. It is to be fixing of nitrogen. In this bacteria, nostoc and Asclepitorium and Anabaena. Okay, Asclepiatoria and Anabaena. So these are to be useful to increase the soil fertilization. To protecting for plants. Means they are all organisms, all bacteria is to be found in the ecosystem, in the underground. They are to be protecting for root systems and different locations. Means this is called a biofertilizers. Means it is to be acting the microorganisms to be given biofertilizers for the plants. Means the harmful pathogens to be killed. What are the organic materials comes from the soil? They are to be supplying for the plants. That is called biofertilizers. Means it is naturally it is to be supplementing for the organic materials. So they will be supplied in different things. They will be supplying different things, and this is also to be supplying for different items. Okay, this all things will be protecting for plants. This is called cyanobacteria. Cyanobacteria is having nostoc and Asclepitorium and Anabaena. This is to be fixation of nitrogen and increase the soil fertilization. Using soil fertilization. Okay, this is biofertilizers. Okay, bacteria is having rhizobium and aspergillum and azotobacter. These three things are having bacteria. These three things has to be found in root system in the plant. The root system it is to be collecting of nitrogen nodules. Means nitrogen fixation. They are collecting nitrogen nodules to supplying for the plants and prevent the pathogens. Okay, then all organic materials also to be supplying for the plant. Next, fungi means it is a mycorrhiza. This mycorrhiza is two types. One is endomycorrhiza, another one is ectomycorrhiza. Ectomycorrhiza found in epidermal tissue of the roots, and epidermal tissue it is to be protecting. Outer layer will be protecting. At the same time, also roots. The roots will be taken in different types of organic materials will be receiving from the soil. So that's time it will be protecting. Why? Means they are entering for pathogens in the root system. That's time this is ectomycorrhiza will be kill the pathogens and to supplying for roots. And endomycorrhiza it is to be found in xylem. Xylem means, so what are the organic raw materials will be supplying throughout the tree? It is to be helpful to using supplying for water, minerals and nutrients. In cyanobacteria means nostoc and Asclepitorium and Anabaena. These are to be found in the soil to increase soil fertilization and to fix the nitrogen. Fixation of nitrogen. This is the biofertilizers. Last thing is coming to, it is called challenges passed by microbes. Challenges passed by microbes means, so we are using different types of antibiotics and medicines to kill the microorganisms or pathogens. That pathogens are to be mutations. Create different types of mutations. So that different types of mutations will be form a super microorganisms. Super microorganisms means it is using in different types of antibiotics and medicines, okay, to kill different types of pathogens. That pathogens are to create different mutations, different mutations. That mutations also to be form a super microbes, means super microbes. That super microbes also to be creating different types of diseases, different types of diseases. That first one is called emergent disease. Emergent diseases. That is called, first thing is AIDS. Second one is SARS. Okay, AIDS means acquired immunodeficiency syndrome. SARS means severe acute respiratory syndrome. Okay, severe acute respiratory syndrome. Okay, this is emergent diseases. It is unfortunately to found these type of diseases. Okay, this type of diseases means, so we are discussing super microorganisms, means so we are using to kill different types of microorganisms. In using antibiotics and medicines, they will create a different mutations. That mutations also to be formed super microorganisms. That super microorganisms will be create emergency diseases. That emergency diseases, the first thing is called AIDS and the second one is SARS. This is called unfortunately to found. Okay, AIDS is acquired immunodeficiency syndrome, severe acute respiratory syndrome. Next, second thing is re-emergent disease. Re-emergent disease. Re-emergent diseases means cholera, and tuberculosis and dengue fever. Re-emergent diseases means, so once upon a time the diseases will be there, but sometimes the diseases will be appear. In some people. Some people have to struggle in cholera, tuberculosis and dengue fever. This is also re-emergent diseases. Re-emergent diseases means, so once upon a time the diseases will be found, but still the diseases will be appear in some peoples. Okay, still the diseases will be appear in some peoples. That is called re-emergent diseases. And one more thing is called bioterrorism. Bioterrorism means the biological scientist will be create a new viruses to release and spread out in all over. That viruses are to be kill remaining all organisms. That organisms are to be kill human beings, animals, whatever it is. That is called bioterrorism. Means the biological scientist will be create a new viruses in man-made viruses to release that viruses to kill human beings. For example, nowadays it is called a corona. Corona will be spread. Means it is man-made virus. Means the biological scientists will be create the coronavirus. It is releasing from the lab. Okay, so it will be create and spread out in fastly to it is to spreading fast and quick and to kill the people. This is also we are discussing from re-emergent diseases. This corona also to be found in previous years. Means that coronavirus it is not dangerous and it is not spreading fastly, and it is somewhat better. But the corona will be create different types of mutations. So they will be very dangerous and fastly. So it is fast to spread out in throughout, and it will be kill the humans, to kill the humans. Okay, the corona is second wave is there. It is the different mutations will be there. The mutations, it is fastly to spread out, in the air, to be to kill the peoples, means the causing organism is respiratory systems and kidneys. They will be spoiled and immediately they will kill. Okay, this corona will be discussing also in bioterrorism and re-emergent diseases. Means the corona will be found in previous years, but that is not a dangerous, and they will be not influenced in any organism in body. Okay, they will come and it go out. Okay, it uses different types of medicines, they will kill. Okay, the corona will be, it is some mutations will be there. Okay, every year it will be different mutations. Every year it will be create different mutation. That mutation in 2019, that is called COVID-19. The mutation is very dangerous. It is fastly to spread in the human beings, to kill that human beings. The bioterrorism will be prevents two things. One is genomics, and another thing is bioinformatics. Bioinformatics means the bioterrorism will be, for example, now we are discussing corona. Okay, the corona will be the leakage for lab, so it will be spread out in all over to kill the human beings. In these viruses, to prevent, using genomics and bioinformatics to make the vaccine. The vaccine will be kill viruses. The vaccine will be kill viruses to implement immunity power and antibodies. Implement immunity power and antibodies, and whenever the corona will be enter, that they will be kill and react. Okay, that is called genomics and bioinformatics. Means bioterrorism. This is also bioterrorism, means man-made viruses. The biological scientists will be create new viruses and releases. The viruses will be released to spread through human beings, and they will be die. Okay, so that is this bioterrorism will be prevent genomics and bioinformatics. To using these two things to find out the vaccines to kill the viruses. Okay, the microbes in human welfare will be completed. The chapter will be completed. We are discussing tomorrow and new topic. Thank you.